I'd like to welcome you to this webinar about exploring protein interactions with UniProt. My name is Clemens Pichler. I'm a curator, a biologist working with the UniProt team. The UniProt consortium provides many different resources, all of which are discoverable through our website uniprot.org. For this webinar, we focus ourselves on the UniProt knowledge base, which is highlighted in the red box on the left of this screenshot of the website uniprot.org. When working with UniProt knowledge base data, it's important to know the difference between the two data sets that the knowledge base consists of. There is the reviewed data set, commonly referred to as UniProtKB SwissProt. This is curated by experts, people like me, who read data from scientific papers and annotate them into database entries, at the same time also annotating sequence features. Sequence features are any sort of data items that can be linked to an amino acid residue or a range of amino acid residues in a given protein sequence. As part of the curation that we do, we also collate isoform sequences into one database entry. The second data set within the UniProtKB knowledge base is the unreviewed data set, also commonly referred to as Tremble. This is maintained automatically, and it does contain annotations from rule systems which automatically transfer annotation from the reviewed data set based on sequence similarity. A lot of those rule systems have human input. We also map experimental sequence features based on 3D structures onto those sequences in the unreviewed set. However, in this set, isoforms are kept separate. Regardless of which part of the UniProt knowledge base you access, you will notice that all the entries contained therein follow a common approach and have a common layout. At the very top of each entry, you'll find the UniProt accession number, which is boxed here in red. This accession number we urge you to use in any scientific publication, as it is both stable and traceable, unlike any other reference that can be used to refer to a UniProt entry. Underneath it, you find a small set of tools to work with, mainly the sequence contained in a UniProt entry. Then there is a header section which contains the basic details about a protein entry, mainly its name, the gene name, and the species of origin, as well as which dataset it's coming from, either the reviewed one or the unreviewed one. On the top left-hand side, you'll find a selection of different views on UniProt data, something we'll come back to later in the webinar. Underneath it, there is a navigation section. Clicking on the individual sections will take you to the relevant section, or you can indeed select which parts of the entry are displayed by using the checkboxes to the left of those buttons. Whatever you select will be displayed in the content area in the middle. If we selected the interaction data for this current example, we would see something like this. You'll notice that the interaction data fall into two categories. There's one subsection called subunit structure and another called binary interactions, both highlighted with a red box here. So it is important to know the differences between these two different subsections in order to be able to use the interaction data as provided by UniProt to their full extent. The subunit section provides information about a protein's quaternary structure, so whether it's a monomer, dimer, or even multimer. It provides binary protein interactions, with the exception of receptor-ligand interactions, which are considered to be an integral part of a protein's function if that protein is a ligand or a receptor, and are therefore listed in the function section of a protein entry. This section also lists host pathogen protein-protein interactions. For example, when a viral protein interacts with a cellular protein during infection. Protein complex interactions are also listed, and if indeed a protein is part of a complex, the composition of the complex is also described. The data is provided in a free text format, and interacting proteins are usually cited with their gene names. This is very important to remember for later on. Data contained in this section can be either experimental, based on data from papers, or added by similarity. If, for example, an interaction between protein A and B was shown in mouse, you might find the equivalent interaction annotation in the entry for the orthologous rat entry by similarity. The binary interactions section is a representation of a dataset imported from the intact database. The database focuses entirely on molecular interactions, and in fact, what UniProt provides is a subset of all the intact data filtered by the experimental support that they have. Intact data only provide binary interactions. This includes homotypic interactions, so self-interactions, as well as interactions with proteins from different species, similar to the host pathogen interactions that I mentioned for the subunit section. All the data in this section are experimental, and there are different views on the data. There's always a tabular view of all the interactions, and there might also be a matrix view, basically a graphical display, but this is only present if there is a manageable size of interactions listed. So if there are too few, there's no point to a graphical representation. If there are too many, it becomes unwieldy, so it's only for the middle ground. Knowing this, what can you expect to find in a given UniProt entry? Well, if there are no known interactions, obviously you wouldn't find subunit or binary interactions at all. Other than that, interactions can be described in either subunit or in binary interactions, or indeed the same interactions can be described in both, and any other combination is possible. This reflects the different annotation priorities and sometimes just manpower of the two different databases that we're talking about here, UniProt and intact. In the end, it doesn't really matter where the information is stored as long as you know how to search for it. Searching for interaction data contained in the subunit section is done like this. Using the UniProt advanced search, you can select interaction as the primary field for searching, selecting subunit structure as the secondary field. It then allows you to enter a gene name to perform a search. So this could be like this, if you'd like to search for interactions listed for BRCA1. Remember that using gene names is important when searching subunit structure. Searching binary interactions is similar, but not exactly the same. Again, you can use the interaction field as the primary one, selecting binary interaction as the secondary field. This time, you don't provide a gene name, but instead a UniProt accession number, like this. This is due to the way data are stored in the intact database, which refers to them by the UniProt accession numbers. Once you've run a search, you will be presented with the results in a tabular view that is common to all the searches in the UniProt knowledge base, like so. If you'd like to jump into the interaction data straight away, the columns button at the top of the table allows you to customize which columns are visible in the tabular results view. And in this instance, you can add a column for subunit structure comments and binary interaction data. And straight away you can see what's available. Sometimes you have both, like in the second entry here, highlighted in blue, or sometimes it's just one or the other. Jumping to a real-world example and looking at the interaction data that it has to offer. As an example, we're using the human BAD protein, which is short for BCL2-associated agonist of cell death. This protein is involved in apoptosis. It's also part of the reviewed data set. Zooming in on the interaction data, we see that for this particular entry, there's both a free text subunit structure comment, which also holds information both at an experimental level. If you view the yellow highlighted evidence tags, as well as information propagated by similarity from another orthologous protein. At the bottom, there's also a binary interactions section, which lists interactions for 12 proteins. You can see the matrix view, and the tabular view, if the matrix view is available, is hidden by default, but it can always be displayed using the link "Show more details" at the very bottom of the screenshot. Within the matrix view, each interaction between two proteins is represented by a blue dot. The color intensity indicates the level of experimental support for this interaction as recorded by the intact database. So the bluer the dot, the more experiments there are to support this particular interaction. Zooming in on the matrix view a little bit further. Highlighted in red is the first column, which basically lists all the interactions of our protein of interest, which is bad, with the other interactors as listed by the intact database. This is equivalent to what you would find as binary interactions in the subunit comment, for example. However, there's more. The bit that's highlighted in yellow details potential interactions, documented interactions, between some of the interactors that are listed for bad human. This is something that goes beyond what the subunit comment would offer, and in that sense is more powerful. Clicking on an individual dot that represents an interaction brings up more information pertaining to this interaction, like, for example, the number of experiments underpinning it, the different identifiers of the involved proteins, whether any of the proteins is involved in disease, here listed in the pathology section, as well as any of the subcellular locations documented for the interaction partners. There's also links to the intact database at the very bottom. Listing pathology or involvement in disease and subcellular location might be of interest here because sometimes one would like to look at protein interactions in the context of disease or in a particular compartment. In order to facilitate this, the matrix view contains filters, both for subcellular location and disease. Basically, these filters are collections of all the annotations for either of the two annotation types that are present in any of the interactors. Selecting one of them filters for the interactions where at least one of the partners has the selected annotation. Multiple selections are possible and are connected by OR. So, in the current example, if we filter for a disease called Leopard syndrome 2, which incidentally is annotated in the RAF1 human protein, it would list all those interactions that involve the RAF1 human protein, because this is the only one that contains this disease annotation in this example. You see that there are 4 other interactions for RAF1 human, in addition to a self-interaction, which you can see in the matrix view. But sometimes knowing about the existence of an interaction between two proteins is not enough. One would like to know which parts of a protein sequence are actually involved or necessary for an interaction to happen. We can investigate this using UniProt data, if such data is indeed available, using a different view of the entry, which is the feature viewer. In this example, PCGF1 human, we have an interaction that's listed for B-core human, another human protein. If we switch over to the feature viewer, we see something like this. The feature viewer is a graphical display of all the annotations that can be tied to an amino acid residue or a range of amino acid residues. Annotations are grouped by different categories in light blue on the left-hand side, which can be folded or unfolded to hide them as you see fit. Zooming in on the domains and sites category, which is the top one, we find that there's a region annotated stretching from amino acid 167 to 255 that apparently is sufficient for interaction with B-core and indeed another protein. That's based on information provided by a paper, which is also listed with its PubMed ID. This information now could help you design a mutant, for example, to knock out the interaction and look for the potential changes afterwards. Turns out that someone's already done this. In another section of the feature viewer, we see that a paper provides a mutagenesis experiment where the change from valine to aspartic acid knocks out and abolishes the interaction between our protein of interest and B-core and indeed this other protein. So to recap, UniProt knowledge base provides interaction data in various boxes. One of them is called subunit structure, another one is called binary interactions. If available, we also annotate relevant amino acid residues as such directly onto the sequence. And both of these bits can be searched using the advanced search facilities that the UniProt website provides. I hope this gets you started in investigating UniProt data with a view to interaction data. If you get stuck, you will find help in our help section. You can also explore the intact database. We have online training courses available to get you started on UniProt in general. There are also training videos on our YouTube channel, and with any other question, you can always contact us using help@uniprot.org. Before we close, I'd like to point out that the effort of providing UniProt and the different resources involves many, many people, and indeed also a lot of funding agencies whose help we'd like to acknowledge. And with that said, I'd like to point out that you can check the EBI training pages for upcoming webinars, and indeed give us feedback and tell us what you think about this webinar and others. Thank you for your attention.